Hello students, myself Dr. Bandana Kumari, Associate Professor, Department of Zoology, Ranchi Women's College, Ranchi. Today I am going to give a brief lecture on viruses. Viruses are very small structures capable of passing its genetic material to a living host cell. A virus is essentially composed of a central core of nucleic acid, which is either a RNA or DNA, surrounded by a protein coat. Some viruses are enclosed within a lipoprotein or glycoprotein membrane called an envelope. The core, protein coat, and if present, the envelope are referred to as a virion. According to Loria and Darnell, 1967, viruses are entities whose genomes are elements of nucleic acid that replicate inside the living cells. Using this cellular synthetic machinery and causing the synthesis of specialized elements that can transfer the viral genome to other cells. If the definition of life is limited to the ability to replicate, then viruses may be considered to be alive when present in a host cell. However, outside their host cells, viruses are merely collections of macromolecules that are in a more or less inert state. Thus, apart from their host cells, viruses can hardly be described as live. Viruses have to really totally on other living cells. Viruses have to rely totally on other living cells for their replication, and thus they are described as obligate intracellular parasites. In addition to being obligate intracellular parasites, viruses also exhibit cell tropism. This means a particular virus has limited range of cell types which can host that particular virus. Cell tropism is facilitated with the help of anti-receptors on the surface of the virus and receptors on the host cells. Cell tropism also determines the species of animal, plant, fungus, or bacterium will be susceptible to that virus. It is for this reason that dogs do not suffer from poliomyelitis and that humans do not suffer from canine distemper. Canine distemper is a disease contracts within dog whereby the symptoms of nose secretion, vomiting, diarrhea, etc. in dogs. For instance, the hemo-agglutinin molecule on the surface of the influenza virus attaches to sialic acid residues on the surface of respiratory endothelial cells. Similarly, a glycoprotein molecule, GP120, on the surface of the human immunodeficiency virus attaches to the CD4 receptor of specific cells of the human immune system. So there occurs a bond between receptors and anti-receptors quoted just now. Bacteria are also susceptible to virus infection, and the viruses that infect bacteria are termed bacteriophages. Now, what is the nature of viruses? What will be the size? The size of viruses ranges from 10 nm to 300 nanometer in diameter. The target animal virus is of 300 nanometer, as for example, smallpox virus. The largest animal virus is of 300 nanometer, as for example, smallpox virus, and the smallest virus, that is foot and mouth disease virus of cattle, BB of 10 nanometer in diameter. On an average, the viruses are 100 nanometer or lesser. To imagine the size of virus, we can also see that virus is 100 to 500 times smaller than bacteria and cannot be seen under light microscope. Only giant virus can be seen under compound microscope. The virus can be seen under confocal microscope using fluorochromes and transmission electron microscope. What will be the organization of virion? Most viruses have relatively simple structure. The basic structure of a virion is the capsid, which encloses only one of the two types of nucleic acids. The infective particle of some plant diseases are devoid of capsids, hence called viroids, which are only a low molecular weight RNA. Capsids are made up of protein subunits called capsomeres. Capsomeres are the building blocks of capsids. Capsomeres are arranged on geometric considerations. The capsid and the nucleic acid core form the nucleocapsid. The large complex viruses often have an additional covering, the envelope, surrounding the capsids. The envelope is phospholipid or lipoprotein bilayer derived from host cell membrane that have been modified by the addition of virus protein. In some envelopes, the proteins appear as projection called spike. The envelope may exhibit pleomorphism, altering the appearance of the virion. There are mainly two types of capsomere assembling: icosahedral and the helical capsid. Icosahedral capsid, there are 20 similar triangular surfaces with 30 edges and 12 corners in the capsid. The number of capsomere is also constant in a particular virus. For example, the adenovirus has icosahedral capsid consisting of 252 capsomeres. Herpes virus has 162 and turnip yellow mosaic virus has only 32 capsomeres. Adenovirus has also a spike protruding from its icosahedral capsid. Now about helical capsid, which resemble long rods with capsomeres arranged around a spiral coil of nucleic acid and is with tobacco mosaic virus and influenza virus. The HIV, rabies virus, and herpes virus have their capsid enclosed by an envelope. Now, what is bacteriophages? Bacteriophages are obligate intercellular parasites that multiply inside bacteria by making use of some or all of the host biosynthetic machinery. Typical phages have hollow heads where phage DNA or RNA is stored, and tunnel tails, the tips of which have the ability to bind to specific molecules on the surface of their target bacteria. The viral DNA is then injected through the tail into the host cell, where it directs the production of progeny phages, often over 100 in half an hour. These young phages burst from the host cell by killing it, that is the host cell, and infect more bacteria. The shapes and forms of bacteriophages have been elucidated mainly to T series of Escherichia coli phages. These are numbered as T1, T2, T3 up to T7. Now, how these viruses multiply or replicate? The reproduction of viruses is usually termed replication. Virus replication takes place in a susceptible host cell, and the series of events that take place from the entry of the virus into its new host to the subsequent release of progeny from the infected cell is termed the virus growth cycle. The duration of this cycle ranges from less than 30 minutes to several days in a small to large animal virus respectively. First of all, viruses must attach to specific receptors on specific potential host cells. They enter through various modes. In bacteriophages, the cell wall of bacteria may be breached by grafting or as a result of direct injection by virus carrying insect vectors. In animals, the entry of virus is either through direct contact or through droplet nuclei, and in the cases of phages, only the virus nucleic acid penetrates the bacterial cell wall by an elegant injection device associated with the phage tail. Based on the relationships with the bacterial cell, phages are of two types: virulent or lytic phages that cause lysis of the host cells, and the temperate phages that may lyse the host, but sometimes their genetic material becomes associated with that of the host cell as prophage and is transferred to daughter cell on division. The bacterial culture which carry the temperate phages are termed lysogenic. Excision of the phage DNA can occur spontaneously or by treatment with ultraviolet light or mutagenic agents which thereafter undergoes for lytic multiplication. The replication process of bacterial virus includes the following steps. Adsorption of free phage. First of all, adsorption of free phage takes place on the surface of host cell. The specificity of adsorption depends on the specific host-phage relationship. For successive adsorption, the phage must collide with the bacterial cell and a bond must be formed between the chemical groups present on both. For example, between Escherichia coli and T1 phage, the amino groups on the viral surface make bond with receptors with acidic group present on the cell surface. These receptors may be located in different layers of the bacterial cell wall. For example, receptors for phase T2 and T6 exist in the lipoprotein layer, whereas those for T3 and T4 are present in lipopolysaccharide layer. Accordingly, the phase surface must also be modified. The bond formation may require the attachment of positively charged cations. Magnesium ions of L-tryptophan can be an example. The T-even phage is equipped with fibrils which get adsorbed to the receptors of cell, leading to a characteristic position of virus at right angle to the cell wall, and the head is sticking out. Some phages attach to the flagella of bacterium. Sometimes mutations occur in receptors which make the cell resistant to viral adsorption. If phages are added in excess, multiple adsorption up to 200 to 300 phages per cell can occur. Now, second step is the penetration of phase nucleic acid inside the host cell. The experiments of Hershey and Chase, who differentially leveled the capsid protein with 35S isotopes and nucleic acid with 32P isotopes, observed the presence of only the level associated with virion nucleic acid, DNA may be DNA, inside the bacterial cell. The bacteriophage tail tip contains an enzyme, lysozyme, which hydrolyzes the mucopeptide layer, a small portion, and after contraction of the tail sheath, the hollow stylus penetrates inside and reaches the plasma membrane. The DNA of the head of the bacteriophage passes through the tail core, that is a stylus, and enters the bacterial cell. The source of energy for driving through the tail core is contained in the sheath of the stylus. Those unsheathed bacteriophages gets energy for driving nucleic acid into bacterial cell from the host supply. Now the third step, intracellular development of phages. Soon after the infection, a bacterial cell, by injection of phage nucleic acid inside the cell, the number of phages is not increased and thus its presence cannot be demonstrated for a short while in the ruptured bacteria. This short period is called the Eclipse Phase. The latent period is the time taken from infection to the appearance of new virus in the medium. Once the viral genome is released from the virion, it becomes available for replication and transcription, serving as template for the biosynthesis of specific products. the synthesis of bacterial DNA stops abruptly, followed by the cessation of its RNA and protein. At first, phage DNA is synthesized at the expense of degraded bacterial DNA. The enzymes required for DNA synthesis are made shortly after injection. These are the so-called early protein. The late proteins includes the coat proteins for the phage, head and tail, as well as phage lysozymes. These are made during the second half of the late period. Genetic recombination, in which phage particles undergo random exchange of genetic material, can occur at this stage. Now the fourth step is the maturation and release of new phages. The fundamental step in virus replication is the production of new molecules of nucleic acid, which are replica of the nucleic acid of infecting particle. The synthesis of viral protein can also take place simultaneously. Maturation includes the assembly of these components, which need several steps. Controlled by genes in the phage nucleic acids, the bacteriophage head and tail are assembled separately from their respective subunits. The virus DNA is then packaged within the head, and finally the tail of the virus is attached to its head. Once a critical concentration of mature virion is reached within the infected cell, they are released into the environment. By a process of cell lysis. In the case of T4 bacteriophage, a virus-encoded enzyme, lysozyme, is synthesized within the bacterial cell, and this causes breakdown of the cell wall, leading to cell lysis. Like other virus proteins, the transcription of the messenger RNA responsible for lysozyme production is under temporal virus control. This means that this is produced during the virus growth cycle at a specific time. The number of bacteriophages released per cell is the burst size. The above cycle, resulting from virus growth cycle up to the release of the bacteriophages through cell lysis, is referred to as lytic cycle. Development of lysogenic bacteriophages or temperate bacteriophages. In addition to lytic cycle, some bacteriophages may have their DNA incorporated into that of the host bacterium. They thus exist in a latent state without causing bacterial lysis. Such state is called lysogeny. The bacteriophages that are capable of undergoing lysogeny are called lysogenic bacteriophages or temperate bacteriophages, and the host bacteria in which they reside are called lysogenic cells or lysogens. One of the most extensively studied lysogenic bacteriophages is bacteriophage lambda, found in Escherichia coli lysogen. The DNA of bacteriophage lambda adopts circular form and can go for lytic or lysogenic cycle. The inserted bacteriophage is called prophage. Most of the prophage genes are silent, as they are not transcribed to form their own proteins by the repressor proteins encoded by bacteriophage genes. Thus, a prophage remains in the latent state within a lysogen, and the bacterial DNA, when replicates, prophage also get replicated, followed by the cell division. Each cell is having a copy of prophage in its genome. Extracellular agents like chemicals, ultraviolet irradiation, temperature, or rare intracellular events act as stimuli that causes excision of the prophase from its host DNA. Once excised, the prophage may initiate a lytic cycle and may give rise to many new virus particles. The example of lysogenic bacteria are Corynebacterium diphtheriae, which carry a prophage which encodes for the diphtheria toxin. Other examples are Clostridium botulism that carry prophages coding for botulism toxin, and Streptococcus pyogenes is having prophage and coding for scarlet fever toxin. After discussion on the replication of bacterial viruses, that on the replication of animal viruses is as follows. The animal viruses have evolved themselves as per the constraints of eukaryotic cell architecture. For example, the poliomyelitis virus can synthesize messenger RNA from their RNA genome template, may act as template. Vaccinia, that is pox virus, can cause DNA synthesis from their double-stranded DNA template in the cytoplasm of the host cell, and the human immunodeficiency virus can cause the synthesis of a DNA copy from its RNA template. Thus, these animal virus replication includes three types: replication of RNA virus, DNA virus, and a retrovirus. First of all, a bond formation between receptors of host cell and anti-receptors of virus takes place. The anti-receptor proteins are distributed throughout on the surface of virus. The cell receptors are generally glycoproteinous in nature. The bond formation causes and configuration change in virus particle, that is, molecular rearrangement on its surface, resulting into uncoating of virus particle and penetration of nucleic acid inside the new host. For example, it is thought that the glycoprotein GP120 molecule on the virus surface acts as an anti-receptor that binds to the CD4 receptors on its target cell surface. This binding stimulates a GP41 of the virus to induce virion fusion with the plasma membrane of cell. The penetration involves three mechanism types: translocation of the entire virus across the plasma membrane of the cell. Second is the receptor-mediated endocytosis of the virus particle, and thereby accumulation of the particle inside cytoplasmic vacuoles. And third, fusion of the plasma membranes with the envelope of the virion. Non-enveloped viruses, poliomyelitis virus, penetrate cells when the entire virus translocates across the cell membrane. In this process, the capsid becomes modified in such a way to allow cellular enzymes to degrade its structure, facilitating the release of the virus RNA genome for translation and the biosynthesis of new RNA strands. Some scientists stresses the mechanism of receptor-mediated endocytosis, and some stresses receptor-mediated pinocytosis, but still no consensus has been released. Enveloped viruses such as influenza virus, herpes viruses, and the human immunodeficiency viruses penetrate cells by fusion of the envelope with the cell membrane. In such cases, the virus envelope remains as part of the plasma membrane of the target cell. The internal constituents of the virus are thus released into the host cell, where nucleic acid is set free. The genome is released from the capsid by capsid proteolysis, and this is achieved with the help of cellular enzymes. The nucleic acid of DNA viruses such as adenovirus, herpes viruses and polyoma viruses, except pox viruses, is replicated in the nucleus of the host cell. Transcription also occurs within the nucleus, and the messenger RNA is transferred to the ribosomes of the cytoplasm for translation into virus proteins. These proteins may be transported back to the nucleus for assemble new virus capsids. In the RNA viruses, replication and protein synthesis take place in the cytoplasm of the host cell. Influenza virus require nuclear factors for their early replication, but rest takes place in the host cytoplasm. In retroviruses, their RNA replicates into complementary, or I should say transforms into complementary DNA in the cytoplasm of host cell and then transferred to the nucleus to become integrated into the host genome. Once certain viruses have assembled their capsids, they be to mature by acquiring their envelope. The envelope is composed of virus encoded proteins and lipids derived from the host cell membranes. Example is the HIV, whereas herpes virus forms their capsids by budding off from the host nuclear membrane. In both the cases, the virus proteins first deposited in the host cell membrane before the acquisition of envelope by the mature virus. After this maturation, they may be released from the host cell by its lysis, and the cycle goes on. So this is all about the virus and its replication. In addition, I have mentioned about certain brief content on the bacteriophages and their replication. Thank you so much.